Hello there, greetings to you. Thank you for joining in to part 3 of our series of classes about the topic the cell. Part 3 is focused on the cytoplasm, and just a brief reminder of the review about part 1 and part 2. Part 1 focused on the introduction to the parts of the cell. We have introduced that the cell is made up of 3 major parts, that is cell membrane, cytoplasm, and the nucleus or introduction that we found in part 1. If you haven't visited that, I kindly request you to first visit those 2 parts and then tune into this part focused about the cytoplasm. Part 2, we discussed the details of the cell membrane, where we found out cell membrane as a lipid bilayer, phospholipid bilayer. I also found there other components, for example, proteins and other forms of lipids. And then we saw its functions, majorly functioning as a selectively permeable barrier membrane. Then we also discussed about the transportation mechanisms that are used in moving materials or substances across the plasma membrane. So now we are focusing on the cytoplasm as the second part of our topic. It is part 3, but it is the second part of the cell. So the cytoplasm is basically a fluid matrix in which other intracellular uh, structures of the cell are embedded, as we're going to see. This cytoplasm is we found different in different cells because different tissues will have different specified functions. For example, muscular tissue should adapt to muscle stress, where we have uh, continuous contractions and relaxations of the muscles. So the cells there have to adapt to those stressful conditions of contraction and relaxation, moving body parts. So the cells there, their cytoplasm will not be the same as the cytoplasm of the cells of the uterus or the cells of the kidney organs, because the others might, for example, the kidney might require an adequate amount of fluid, of water within the cell, unlike the muscle tissue. So the cells there will have the amount of fluid in the muscle tissue cells will not be the same as the amount of fluid in the cytoplasm of the cells found in the maybe uterus or the kidney. But it is mostly the cytoplasm. It is mostly composed of water. So to introduce something about the organelles or the structures found within the cytoplasm, you may also refer to them as cytoplasmic organelles or the cytoplasmic structure. So these have complex characteristic shapes, but the shapes are organized in some way to suit particular functions that are specific. They have unique shapes, as we are going to see, and each organelle that we are going to see performs different function independent of the other, but in most cases they coordinate with one another to fulfill the overall function of the cell, which later on fulfills the overall designated function of that particular body organ or tissue. The cytoplasmic organelles are important or essential for the normal functioning of cellular structure and activities. And they are categorized into two major groups or types. We have the membranous organelles and then the non-membranous organelles. You may also refer to them as membrane-bound organelles or structures and then the non-membrane-bound organelles structures. The membranous organelles have a membrane surrounding their internal components or structures, and that membrane that surrounds those structures is similar to the composition that we saw in the plasma membrane. For purposes of these organelles easily interacting with the plasma membrane in terms of transporting particular substances across the plasma membrane in or out of the cell. And then the non-membranous organelles, those ones, they don't have a membrane surrounding the internal components that we find there. But however, they in most cases they have a protein coating around them, so they find a way of interacting with the plasma membrane, either first interacting with the membrane bound organelles, and then they get that coating from the membrane bound organelles, and later on they interact with the plasma membrane, or they find a way of interacting with the plasma membrane, for example, through channel proteins or integral proteins, so they can always be transported across the cell membrane. So those are the two major groups or types of cytoplasmic organelles that we are going to discuss. So to start off with the membrane bound organelles or the membranous organelles. These ones include the first one is the endoplasmic reticulum, which is of two major types, that is the rough and the smooth endoplasmic reticulum. Uh, the rough endoplasmic reticulum has ribosomes, as we're going to see, on its surface. So it has ribosomes fixed onto its surface for particular functions, which mainly include synthesis of proteins for exportation, either extracellular or intracellular. But it is functioning in protein synthesis, because as we are going to also see, ribosomes are the sites of protein synthesis, and if we find the ribosomes being fixed onto this rough endoplasmic reticulum, then the proteins that will have been synthesized from the ribosomes or at the ribosomes will then be transported by the rough endoplasmic reticulum and then exported to either internally still within the cell or outside of the cell. The rough endoplasmic reticulum is also responsible for the synthesis of peroxisomes. Peroxisomes are membranous organelles that we are going to see, whose functions are mainly in dealing with toxic substances that enter the cell. So in detoxifying cells, that is where we mainly find the peroxisomes doing their work, performing their roles. So that is it with the rough endoplasmic reticulum. They have ribosomes fixed onto their surface membranes. So the smooth endoplasmic reticulum, on the other hand, for them they don't have ribosomes fixed onto their surface membranes, and their roles will thus uh, differ from those of the rough endoplasmic reticulum. While the rough endoplasmic reticulum their role was to synthesize proteins for transportation. These ones, they are the smooth endoplasmic reticulum. Their major role is to synthesize lipids, and we also find them functioning in detoxification processes. their major role is to synthesize lipids, unlike the rough endoplasmic reticulum. We also have to note that this organelle or structure is continuous with the uh, nuclear membrane. So it is a continuation of the plasma membrane, of the nuclear membrane. So that is the endoplasmic reticulum, where we have these bead-like structures in brown color. Those ones are the ribosomes fixed onto the surface membrane of the plasmic reticulum, and we see the nucleus here. So this membrane of the plasmic reticulum is continuous from the membrane of the nucleus. So we are likely to find it close to the nucleus because it is continuous from the nuclear membrane. These foldings make spaces, and those spaces are known as cisternae. So uh, in these spaces, we find the transportation of the proteins that will have been synthesized from these amide-like structures, the ribosomes. So after protein synthesis that takes place on these ribosomes, within these spaces is where we shall see the vesicles of the proteins being transported in form of vesicles, then released at one end. So we also see this continuation of the nuclear membrane, which does not have the ribosomes, and we have said that that one is the small endoplasmic reticulum. So those are the functions of the endoplasmic reticulum. Just to review, it can function in transportation, synthesis of particular organelles, molecules like proteins, if I don't have ribosomes on the surface, storage of certain molecules, and then so it is the endoplasmic reticulum can function as a temporary storage of particular molecules, macromolecules, for example, lipids. Lipids can be found temporarily stored the within the cisternae, those spaces of the smooth endoplasmic reticulum, because the smooth endoplasmic reticulum uh, functions in the synthesis of lipids. So we can find a temporary storage of those newly synthesized lipids within the smooth endoplasmic reticulum, those internal spaces, which we have called or referred to as the cisternae. And then on the other hand, for the rough endoplasmic reticulum, we can find a temporary storage of proteins within uh, within those structures, the cisternae, those spaces within uh, the rough endoplasmic reticulum. And you can also see or find the endoplasmic reticulum functioning in detoxification reactions, where we find the smooth ER detoxifying drugs and alcohol, because the lipids that are being synthesized in the smooth endoplasmic reticulum to be packaged will help form packaging vesicles of these drugs and alcohol molecules that have been obtained from alcohol that was taken by these individuals. So in doing so, they are functioning as detox. They are functioning in detoxification processes of the cell. Those are the functions of endoplasmic reticulum, both smooth and rough. To continue the membranous organelles, we have structures or organelles known as peroxisomes, and we have said these ones are synthesized by the rough endoplasmic reticulum, and they are vesicles in nature, uh, which utilize oxygen in their detoxification processes. So they are mediated by specific enzymes. So we are likely to find enzymes being enclosed within these structures, and then those enzymes function in catalyzing particular reactions or catalyzing particular reactions within the cell. So when we have a molecule that has been taken up by the cell after being transported across the plasma membrane, the enzymes that we find in these peroxisomes will then be released while inside the cell, will then be released, and then they interact with those vesicles that were formed after the endocytosis processes, and then these enzymes from the peroxisomes will carry out their roles of catalyzing those reactions after they interact with those molecules that have been received within the cell. The peroxisomes are organelles that are mostly abundant in the liver because the liver is uh, the main organ in which detoxification takes place. So we find the peroxisomes numerous in the liver cells. So that is the structure of the peroxisome. Inside it, we find components such as enzymes. So another organelle under the membrane bound organelles, that is the Golgi apparatus. This one functions in modifying or storing and sorting material that has been obtained from the rough endoplasmic reticulum. So after materials have been synthesized from the rough endoplasmic reticulum, which are usually proteins. Okay, so protein synthesis took place on the ribosomes, which were found on the the membrane of the rough endoplasmic reticulum. Then those proteins were transported to the cisternae, those spaces of the rough endoplasmic reticulum, then released as a vesicle, which vesicle is then which vesicle then interacts with the surface membrane of the Golgi apparatus for modification purposes or for storage or certain processes that require sorting out certain molecular components of the products that was obtained from the rough endoplasmic reticulum. So the Golgi apparatus has two phases: the receiving phase, which is the seize phase, and the shipping region, which is the trans phase. Uh, the Golgi apparatus also functions in producing or synthesizing lysosomes. So these lysosomes also, in the end, function in autophagy and autolysis. So after the lysosomes have been synthesized from the Golgi apparatus, they will then go ahead and carry out their roles as in autophagy, which is the removal of old organelles from cells, or autolysis, which is the destruction of old cells. So that is the Golgi apparatus. They are having two phases. We have this receiving region where we see a vesicle being received on this side, and then we see this shipping region where we see a vesicle being released here, vesicles being released here, then vesicles being received this side. So within these spaces, the systemic spaces of the Golgi apparatus, we see transportation or temporary storage, where those processes of modification take place and sorting of those particular molecules. All those will take place within this systemic space, and then later, after all the necessary biochemical reactions have taken place, the transportation vesicle will then be formed and released from the Golgi apparatus for further processes. It can be released as a vesicle or can be released as a vacuum. So this is a continuation of still the Golgi apparatus, how it functions in the cell. So it can receive particular vesicles from the endoplasmic reticulum, as you see here. And once it receives from its receiving region, it processes them appropriately and then releases. It can release, and those vesicles stay within the intracellular component of the cell, or the vesicles released from the Golgi apparatus can fuse directly with the plasma membrane, and then the plasma membrane releases the necessary components that require to be released from this cell. So realize something can be produced within the cell and undergoes about four stages of processing, multiple stages of processing. For example, protein synthesized at the ribosomes, and then they send their product, the plasmic reticulum. The plasmic reticulum transports it and forms the vesicle, which sends it to the Golgi apparatus. Also, the Golgi apparatus modifies whatever it has to modify and then fuses with the plasma membrane, and it is exocytosed. So something can undergo multiple stages of processing within the cell. So those are the lysosomes, organelles synthesized by the or from the Golgi apparatus. The other membranous organelle of major interest is the mitochondrion. If you are referring to many, then they are called mitochondria. So they have the characteristic of being double membrane bound. So they have their membrane is a double layer kind of structure. They function in synthesizing energy form of ATP. Another point to note about their double membrane is that their outer membrane is smooth, but the inner membrane is folded. It is folded as you see, but their major role is to synthesize energy in the form of adenosine triphosphate, which is abbreviated as ATP. Commonly, they are known as the powerhouses of the cell because they are responsible for giving this cell energy, producing for it, synthesizing for it energy that it will use in different biochemical processes. So that is the mitochondria structure. The inner membrane is folded. It is folded into, and those spaces that are formed after folding are called the cristae. The spaces in the Golgi apparatus in the endoplasmic reticulum we are called cisternae. These ones are called cristae. Yes, the spaces are called cristae that are formed after the foldings of the inner membrane. Uh, within within the intracellular compartment of this organelle, the mitochondrion, we find the fluid matrix there. We just refer to it as the matrix, but we find the fluid there, mitochondrial fluid there. Inside. And on the surface of the inner membrane, we find certain mitochondrial enzymes. We well know that enzymes are protein in nature. So we find there certain mitochondrial proteins, form of enzymes that are responsible for particular biochemical processes of synthesizing the ATP and other processes. It's not only ATP. So that is the mitochondrion. So to start off, the other group of cytoplasmic organelles, which are the non-membrane bound organelles. Those ones, as we have already heard, they lack a membrane, so they are not surrounded or lined by a membrane. They are usually made up of protein coating. So what they are surrounded is usually of a protein nature, protein coating. So they include those three major types of organelles or forms of organelles. We have the ribosomes, cytoskeleton, and then the centrosome. Ribosomes can either be found free within the cell cytoplasm or found fixed onto the rough endoplasmic reticulum. The other forms of non-membranous organelles, which are the cytoskeleton, can be found in three major subtypes, that is the microfilaments, intermediate fibers, and then the microtubules. And we're going to see the differences between all those. The other one is the centrosome, and it is usually arranged in structures called centrioles. So start off with the ribosomes. These are small, dense, uh, granule-like structures. So refer to them as dense granules. They are small, made up of protein and then RNA. RNA is ribosomal nucleic acid. So that type of nucleic acid, you have two types of nucleic acids in cells. You have DNA and then RNA. But for the ribosomes, we find that the RNA is their major nucleic acid. It's the major nucleic acid that we find associated with them, in addition to the ribosomal proteins we find here. They are a site of protein synthesis, so that is where we expect all our proteins that are produced from the cell to be synthesized. So yeah, that's important within the cell. That is their major role, protein synthesis. Each ribosome has two subunits, that is a small subunit and a large subunit. And usually the large subunit is about 2 times larger than the small one. So the small one is formed as about a half the size of the large subunit. So they are always arranged, you may call them kind of pairs, the small and the large subunit, but they are fused together. That is a ribosome there. They are not kind of pairs, but something fused together with a small and a large. So in the end, you have those two structures. They are functioning together. The cytoskeleton has another structure and a non-membrane bound organelle. So the cytoplasm is mostly composed of filamentous types of proteins, protein filaments. Okay? So it helps give the cell shape because these filaments will always function in providing the support to this cell, so that this support, they are like a network of filaments within this cell. So this network helps strengthen the cell so that it does not easily shrink or lose shape as it is carrying out its biochemical processes, because fluid is always saturating within this intracellular component of the cell. So as the fluid is saturating, we don't find cells forming false structures because of the movement of the fluid. So it is the role played by the cytoskeleton to maintain that shape. But we said we have several types of cytoskeleton, as we're going to see. So cytoskeleton is also important in coordinating cellular movements, like how I've been explaining. For example, in muscle tissue. Those cells there are always contracting and relaxing. So they have a lot of cytoskeleton within their intercellular compartment. So we expect those different types of cytoskeleton to always be coordinating in terms of their function so that these cells don't tear or get torn. When they are torn, what we call muscle tearing, it means the cells were damaged, they were over stressed by either the contraction of the muscles or the uh, relaxation, so they got torn. So we say there are three major categories of cytoskeleton, that is microfilaments, intermediate fibers, and microtubules. What are these? And the microfilaments, you can see they have, it is like a double layer of coiling, so two layers but coiling over each other. Intermediate filaments, there are multiple coils around each other, arranged in some pattern. So they are like a bundle of filaments. Then the microfilaments are like a pair of filaments coiling over each other. Then the microtubules are like a pair but not coiling over each other but coiling alongside each other. As you can see, this pale blue and deep blue, they are coiling alongside each other but not over each other. So that is the difference between those three major types of cytoskeleton that we find within the cell. Microfilaments, they are a pair of filaments or fibers, but they are coiling over each other. Intermediate filaments are multiple fibers, but the coiling is over each other still, but they are multiple. Microtubules are a pair coiling alongside each other. So those are the differences between those types of cytoskeleton. Centrioles and centrosome are other non-membranous structures that you can find within the cell. What are these? Centrosome is an area that is close to the nucleus. Uh, it's an organization site for microtubules. The microtubules that we have seen above, those are pair of filaments coiling over each other. Those green ones there. So they are organized at the sites that we know as centrosomes. So that site is close to the nucleus. So that is where they are organized and then later released into uh, the cell, the cytoplasm of the cell. Then the centrioles, these ones exist as a pair in the centrosome. So in this area close to the nucleus, we also find the centrioles existing as a pair. They are arranged perpendicular to each other, so almost at 90 degrees. So that is being perpendicular to each other. Arranged as 9 sets of microtubule triplets. So we have 3 arrangements times 9 or 9 times. They are important in cell division and spindle fiber formation. So what are these centrioles? Yeah, centrioles are 3 pairs. They are a triplet. So 1, 2, 3. That is a triplet, but arranged in 9. Uh, 1, 2, 3, 4, 5, 6, 7, 8, and 9. They are arranged in 9 sets. So a set of 9 triplets. That is a centriole. But these triplets occur arranged perpendicular to each other. So you realize they have kind of 90 degrees in between each other. But the overall pair again is also arranged perpendicular to each other. You have a pair of 9 sets of triplets, but this pair is also perpendicular to each other, forming about 90 degrees somewhere. Yeah. So those are the centrioles and centrosome. Centrosome is an area close to the nucleus where we find the centrioles. So cells can have other appendages on them, and these appendages are usually extending from the surface of the cell membrane or the plasma membrane. There are mainly three types of appendages that exist amongst cells, that is microvilli, cilia, and flagella. So they are just appendages that are extensions from the plasma membrane. So what are these? Microvilli, you can find certain cells such as the ileum, the cells of the small intestine. They have that characteristic of having microvilli for particular purposes, usually purposes relating to absorption of nutrients from food that has been ingested. Their characteristic morphology, they are short extensions from the surface of the plasma membrane. Cilia usually occurs in numerous numbers, and they are also usually short extensions but occurring in numerous numbers, unlike the microvilli. Those ones we can find them in the lining of the nostrils, an organ of the respiratory system. So we find the cells having cilia there. They help in the movement of materials along the surface of those cells, or they block particular unwanted materials from going into delicate parts of that system, respiratory system. For example, they block dust from entering the lungs, the bronchioles of the lungs. Flagella is another appendage that we can find as an extension from the surface of the plasma membrane, and an typical example of cells with flagella are the spermatozoa cells, sperm cells. Those ones have a flagella, and the major function of flagella is for movement of uh, those cells. So those are the depictions of microvilli, cilia, and the flagellum. So part A is having microvilli. Part here we are seeing cilia, and then here we are seeing flagella. That's the end of part B, which is what was the second part of the cell, known as cytoplasm. And be blessed wherever you are, and yeah.